So we talked about the need for surveillance. Now, if you think about it, what is the job of the hospital laboratory? So as patients, people go into a hospital and invariably someone draws their blood. That's what we do in hospitals when people come in with fever and we're trying to figure out what type of infections they may or may not have. What happens to that blood? That blood gets sent to a laboratory, most commonly in the hospital. Sometimes they outsource it, but usually in the hospital. And that hospital's job, that laboratory, is to take that blood and ask whether any bacteria is there. Because in general, your blood should be sterile. So they grow it. And they ask whether is something going to grow. And something does grow, then the question is, what is it? And then they identify it. And then the next question is, if it is a bacteria, then the question is, what can I treat it with? So those are the jobs of the hospital lab, and the faster they can identify the bacteria and tell the treating physician what drugs I can use to treat the patient, the better job they do. It normally takes on average 48 to 72 hours before the physician has that information in their hands. What do you do during the first 72 hours when a patient's in a hospital? Well, most of the time a doctor is treating empirically from the seat of their pants. It's based on experience, it's based on presentation, but they're basically giving broad spectrum antibiotics to try to think that they're covering whatever this infection is, if it is an infection, and I'll know soundly within 72 hours. So that's what happens in a hospital laboratory. That's their job. Research laboratories like mine, we can ask the next question that once you have a bacteria that is drug resistant, we can analyze it further and ask questions like surveillance. Do they have the MCR1 gene? Do they have other resistance mechanisms? And in our case, we're not trying to impact on the patient's treatment. We're trying to understand from a societal point of view and a public health point of view, are there lots of these strains, not only in hospital A, but since I can work with multiple hospitals, we have a sense of what's going on in a city, in a community, in a state, by comparing isolates from different sources. And when we recognize that, oh, these three hospitals seem to have the similar problem, then we're starting to understand that surveillance has been telling us that yes, they're spreading and there's bacteria now that are growing in numbers, and this is the type of data that we can generate with the thought that we potentially could have a problem, and that's exactly what we need to do with the MCR1 gene. And as other resistance genes become identified, we can at least start doing the surveillance studies and work with hospitals to provide the information that they can use, not necessarily to treat patients, but more infection control and trying to control the number of these bad bugs in their hospital.